Good morning, everyone. Good morning, ladies and gentlemen, and apologies for the delay. Welcome. My name is Dr. Khawla Ali, and I'm the Master of Ceremony for this symposium. First of all, I'd like to thank you for joining us today in this online symposium on the collaborative research on COVID-19 in the Kingdom of Bahrain, held under the patronage of His Excellency Lieutenant General Dr. Sheikh Mohammed bin Abdullah Al Khalifa, Chairman of the Supreme Council of Health. Next slide, please. Please note that this event is being recorded. We will be holding a Q&A session after the presentations using the dedicated Q&A function of this event, which is available in the top right-hand corner of your screen. Please use this function to share with us your queries. Next slide, please. Today's program will be composed of the following. Our guest of honor will be delivering a speech and welcome notes at a later time. So we will start out with a favipiravir clinical trial study overview, followed by an overview of two COVID-19 research projects. First, the convalescent plasma clinical trial, and then COVID-19 and sickle cell disease, followed by a talk on the efficacy and safety of hydroxychloroquine in COVID-19 patients, followed by COVID-19 breaking down a global health crisis. And we will finally end with the Q&A session. Next slide, please. Next slide, please. Now I would like to invite our guest of honor, His Excellency Major General Professor Sheikh Khalid bin Ali Al Khalifa, Director of the Royal Medical Services and Chair of the Research Committee of the National Task Force for Combating the Coronavirus, COVID-19, to deliver the welcome notes of this symposium.
Good morning, all of you, to this meeting, and I wish you a warm welcome to the Cooperative Research Symposium on the COVID-19 in the Kingdom of Bahrain, and which is held under the patronage of His Excellency Lieutenant General Sheikh Mohammed bin Abdullah Al Khalifa, the Chairman of the Supreme Council of Health. And as you know, this symposium is a result of cooperation between the National Task Force Combating the Coronavirus, COVID-19, and National Health Regulatory Authority and RCSI Bahrain. As the COVID-19 pandemic continues to affect everybody worldwide, we have been living through a challenging and momentous time. The Kingdom of Bahrain faces a challenge of the pandemic in a protective and assertive manner, which led to success of containing the disease and mitigating its impact, while other countries unfortunately witness a deterioration of the situation. Under the leadership of the Government Executive Council, chaired by His Royal Highness Prince Salman bin Hamad Al Khalifa, Crown Prince and Prime Minister, we were able to place and implement stringent regulation, which is gain support from the health professional and the people of Bahrain, who demonstrate a great responsibility and determination to fight this pandemic. The Kingdom of Bahrain outstanding response to the pandemic has been revered and acknowledged globally, notably by the World Health Organization Director General, Dr. Tedros, who praised our comprehensive approach to tackling the pandemic. A clinical study where an opportunity we have taken to shed the light on the spread of the disease, its impact and specific population and possible treatment. I hope you find today's symposium, enriching its lighting some of the advances made in COVID-19 disease, research and guideline implementation within Bahrain. As chair of the research committee of National Task Force for combating coronavirus, COVID-19, would like to take thanks to my colleague in their excellent work, and especially Professor Manaf Al-Qahtani and Dr. Abdul Karim Abdurrahman, who will be presenting some of their research today. I would like to thank also the National Health Regulatory Authority, the Supreme Council Health, and Ministry of Health in their continuing cooperation. And finally, I would like to thank RCSI Bahrain and its particular Professor Steve Atkin for support and providing with the clinical trial. I would like forward to hearing our guest speaker today to present the research finding. And thank you very much.
Thank you, Your Excellency, Major General Professor Sheikh Khalid bin Ali Al Khalifa, for launching the symposium. Next slide, please, if we can move forward. Thank you. First of all, I would like to introduce our first guest speaker for the day, Professor Stephen Atkin, Head of Postgraduate Studies and Research at RCSI Bahrain, to share his research on the favipiravir clinical trial study.
Carla, thank you very much indeed. Esteemed guests, ladies and gentlemen, I'd like to talk about the favipiravir clinical trial study and give you an overview. Next slide, please. As this audience well knows, COVID-19 disease is caused by the severe acute respiratory syndrome coronavirus 2. This has resulted in a serious pandemic with marked public health and economic sequelae. And as of today, there's over 90 million cases, leading to over 2 million deaths. And currently several vaccines are existing. They're being rushed for approval across the globe. Next slide, please. The coronavirus has a spike protein that interacts with the angiotensin converting enzyme 2, transmembrane metalloprotein. And once bound, it allows the virus to gain access into the cell. Next slide, please. Whilst many patients may actually be asymptomatic, the common symptoms that these patients actually have include fever, cough, breathing difficulties, loss of taste. However, for those patients with diabetes, obesity, hypertension, and cardiovascular disease, they are more at risk of developing pneumonia, severe acute respiratory syndrome, development of a coagulopathy, and that may lead to death. There are a number of candidate drugs which have actually been used in the treatment of this disease, and these include chloroquine and hydroxychloroquine, which alter the acidic environment of the lysosomes that then prevent viral RNA release. There's lopinavir and ritonavir, which are protein. Could we go back forward, please? Thank you. Which are protease inhibitors. There is tocilizumab, which is an interleukin-6 receptor blocker that prevents interleukin-6 binding to the IL-6 receptor and thereby preventing the inflammatory process. And there is remdesivir, favipiravir, and ribavirin, all of which inhibit the RNA-dependent RNA polymerase. Next slide, please. Back one slide, please. We here at RCSI are working with the COVID-19 task force, undertaking the clinical trials to actually start looking at fighting this disease. And we're actually achieving this by the employment of our alumni who are placed in the COVID centers and who are doing a magnificent job of actually undertaking the clinical trials, one of which you will hear very shortly, Professor Manaf with the convalescent clinical trial, and of course the favipiravir trial. Next slide, please. So favipiravir is an antiviral drug that was developed in Japan. It's a pyrazine carboxamide derivative with a very wide spectrum of antiviral activity, including that of influenza, for which it was developed, West Nile virus fever, yellow fever, foot and mouth disease, as well as other flaviviruses. And as I say, its action is through inhibition of viral RNA-dependent RNA polymerase. A report that was recently published comparing favipiravir with lopinavir and ritonavir suggested that favipiravir was superior in the prevention of disease progression and viral clearance. Next slide, please. So the primary objective of this favipiravir pilot study was to compare hydroxychloroquine with favipiravir and standard care where patients did not receive any drug treatment in a randomized open-label clinical trial. And the difference between the groups would then allow an effect size to be determined so that we can then do a definitive clinical trial. Next slide, please. As I say, we're supporting this trial with 5 RCSI alumni in the COVID-19 centres, and we are randomizing 50 patients to standard therapy, 50 patients to hydroxychloroquine therapy, and 50 patients to favipiravir therapy. And as of this date, we have over 50% of patients recruited. And we're supporting this clinical trial data using a novel electronic case report form, which has been developed by RCSI Bahrain. Next slide, please. This electronic clinical report form allows us to enter the data for each individual patient into a palm-held device that can then be checked for its accuracy. And then, next slide, please. The data can be sent to the central consolidation area, which is an RCSI Bahrain. And then when the data is all complete from the clinical trial, it can then be sent directly to the statistician in an extract file in order that we can actually analyze the trial expeditiously. Next slide, please. So in summary, RCSI is supporting the COVID-19 task force through sponsoring the clinical trials, providing clinical trial staff to undertake the clinical trials, introducing technology to simplify and implement the clinical trials, collaborating on data analysis, collaborating on data interpretation, and collaborating on the dissemination and publication of the research results. Thank you for your attention.
Thank you, Professor Stephen, for the talk. Now I would like to introduce our next guest speaker, Professor Manaf Al-Qahtani, infectious diseases consultant and microbiologist at the Bahrain Defense Force Hospital and member of the National Task Force for Combating the Coronavirus, COVID-19, to present his overview of 2 COVID-19 research projects.
All right, thank you, Khawla. Good morning, everyone. Before I'm going to start, I really would like to thank Professor Sheikh Khalid. He has been with us for more than almost a year now with this pandemic, and he's truly a leader. Despite the busy schedule and despite the country has been affected massively by the coronavirus, he still encouraged us and he established this research committee, which actually established back in April of 2020. I'm very glad to say, despite all the amount of efforts that people were really engaging in fighting this virus, his true word for us has been always is to convert the current policies into publications. And we have been taking that on our shoulder, and we have actually received 78 manuscripts in Bahrain. And from the 78, I'm very happy to say at least the one that I have a direct hands-on. Some of them are with RCSI, but all of them actually through the BDF hospital personnel, is actually we have completed 19 researches, all related to COVID-19 over 9 months. And I think this is truly an achievement for the Kingdom of the Bahrain. Of those 17 manuscript, we have around 9 has been already published in the journal. Professor Steve Atkin has been very supportive for us during this pandemic. NHRA has been wonderful in actually expediting all the research application, not just for the RCSI and BDF, but all across the kingdom. So we're talking about almost 78 manuscript. The one that I know, at least the one that I'm supervising, the 17 has been completed. And I'm just going to go really through them very quickly in the next slide. So the idea of research, in fact, and again, that was something that has been always in the mind of our leaders, is that we really wanted to use the Bahrain data. You all know COVID-19 has been something new. If you go to the literature back then in February, there's really very little to find about, even in terms of the policy. Bahrain took that lead since the beginning of January 2020, and we have actually set up a research committee where we had members from different sectors in the kingdom, and with a partner with the NHRA and RCSI as well. So the whole idea from all of this work is actually to have our own data and convert them into policies. So every single policy you're going to see here in the Kingdom of Bahrain, it's actually purely related to Bahrain data. And that has been put Bahrain actually in the top leading countries in terms of policy making. We have shared our policy with different GCC countries, and I'm very glad to say that we have shared it with Mexico, we shared it with the UK group as well, and when it comes for the quarantine, testing with the antigen test, and so on. So there are so many things to say, to be honest, when it comes for the research, but I know we are limited with the time. So I'm just going to go briefly through all the papers that we've been working, or sorry, all the research projects that have been working. So the one that have been published so far are 9. The first one is sickle cell disease in Bahrain, which I'm going to talk about it in a few slides. Again, the whole idea from the sickle cell disease, Bahrain is a well-known country that we have around 1% of population being affected of sickle cell disease. So the question definitely, will those be a threat? So we are actually anticipating pure in advance if sickle cell disease would be a problematic, and we're going to find that when I reach the sickle cell disease. The next one, which has been in collaboration with the RCSI, a very fantastic question, which was the prevalence of asymptomatic and symptomatic COVID-19 in the cohort of quarantine subject. If you go back to April and March, when we had the 14 days policy here in Bahrain, and again we had the question about the role of screening at the airport. Does the screening of symptoms at the airport would be helpful or not? And again, you're talking back then, we have limited amount of information. We felt like influenza and like SARS-CoV-1 that those symptomatic individuals would be the only infectious. And when we published that paper at the International Journal of Infectious Disease, basically it shedded to a very interesting information. For example, we have learned that asymptomatic were more prevalent than symptomatic in spreading the virus, especially at the passenger arrival. We have learned even passenger were not the major risk in introducing the coronavirus. It is an element, but was not the major one, which we're going to allude into the other manuscript. The interesting part, actually, we found that very few of those symptomatic were transmitting the virus in comparison with the asymptomatic. So the rule of this, or the bottom line, testing upon arrival has been shown, even back in April, has been shown to be of a great value. And that's why Kingdom of Bahrain started to continue in testing every single person that comes in at the airport, even if it has been tested somewhere else. We have been taking that upon our shoulder in testing all the passenger. Just a reminder, we have never shut down the airport. We have reduced the passenger across the pandemic. And again, that result have been actually shared with the WHO, which I'm going to go into the other manuscript. The other one we talked about, or actually we worked on, it's what was the serological testing. Again, this is back in April when we used the rapid serological testing, which actually showed of no clinical utility in spotting. The sensitivity was less than 37%. And again, I just want to remind, this is not the quantitative antibody testing. This is the rapid antibody test, which we have many literature that support the less value of using those tests in capturing COVID-19. The third manuscript that we worked with RCSI, again, was about the quarantining arrival travelers, balancing the risk and the benefit. And again, if you are in the middle of the pandemic and you want to balance between the economy and the security of the public or the safety of the public, we felt that with the quarantine 14 days, it actually had tremendous negative impact on the individuals. It has a huge psychological impact. We have tested that, and we have again proven, and that has been published in the Tropical Disease Travel Medicine Journal, very well-recognized journal. It actually showed that testing again upon arrival has been more important than the quarantine, and that's why we have lifted the 14 days quarantine, and we have put the 10 days rule in repeating the exit swab, because we have found majority of the people, if they are tested negative upon arrival and let's say they are harboring the virus, it's actually within the 7 to 10 days that they're going to show of the virus. And this is what we have done. Every negative person during the study, we've been doing the PCR every 2 days. I think the number of people were 7,000, if I'm not mistaken. And again, it showed that 10 days was the good timeline that we have selected. And again, this is back in June when we have put this policy. And you're all aware, CDC in November have announced that 10 days actually is as good as 14 days. Again, to give you this kind of an encouraging that if you have a data and it's a value, you really have to utilize it. And we have proven again that the 10 days was a perfect timeline to end the quarantine. Recent advances in vaccine and immunotherapy. Again, this is a group that we work between Bahrain and Saudi Arabia. That was just a mini review about the role of the vaccines. Oh, by the way, I have sent to Celine from the communication office all the manuscript with the hyperlink that you can have direct access to them if you like. Then we reached about the use of radiological imaging. That was a collaboration between us and RCSI students. I think they are from senior cycle. Again, we published that at Future Microbiology. We looked at, it was more of a systemic review. We looked at the role of CT in making diagnosis of COVID. If you remember back in February when China has used just for 1 day CT as a guided in making diagnosis of COVID, and we have seen the amount of the cases, it actually tripled, and next day they have removed from their definition the CT. So CT was a good to make to help you in making the diagnosis, but should not be used as a screening to make a diagnosis of COVID-19, especially when you have other viral illness like influenza, where there's no way you can make a distinction between COVID-19 and other viruses. The efficacy and the safety of hydroxychloroquine. This is a topic has been hot for several months. We had some clinical, benefit of the use of hydroxychloroquine. But as you know, as a science, you cannot use just the clinical perspective. We have seen some good outcome in terms of discharges, but was it really from hydroxychloroquine or other co-founders? And this is what we have proven. This is the largest multi-center trial, at least in the region that I am aware of. And we have looked at the value of hydroxychloroquine over 6 months. And again, it did not show, like with any other literature, did not show any clinical utility or benefit from this drug. In fact, the risk of hypoglycemia tend to be very high in elderly, and therefore we have actually removed this drug from our protocol. We've talked about the mitigation in aviation. The next two publications, this was in collaboration with the WHO. So Bahrain now joining the group called Guidelines Development Group, GDG. Those are basically involving in putting all the policy regarding airlines, aviations. Those are now online in the WHO, and we have shared our quarantine policy, and we have shared our testing protocol, and we have shared with them our antigen test, which is the rapid antigen test, as a way to use it in the coming months with the COVID-19. Next slide. Those are the preprint papers that we have been submitted, and also along you're going to see the name of the journal that we are just waiting for their acceptance. Those are 8 manuscripts. Again, just go through them very quickly. It's about the blood group. I'm pretty sure you've been hearing a lot about the effect of blood group. Can you use blood group as a biomarkers in detecting the severity of COVID-19? And the claim at that time came from initially from a Chinese, then from a Netherlands. They have claimed that if you have a blood group A or AB, you tend to have more severe course of COVID-19, and that was linked due to clotting formation. Other studies have shown that this is not the case. In Bahrain, we said let's just look at the Bahrain population. Since again we started in admitting every single patient regarding their symptoms to a COVID facility, the whole idea at that time, we wanted to study the clinical course of this disease just to give it some background, and I think that was an excellent idea that been implemented, because we were able to have the blood group back then since the first case in February, and all what we found, because the majority of Bahrain, or at least those who are enrolled in this study were group B and O. We had very few group A, but we could not see any significant correlation between blood group and severity. In fact, those who had group B tend to have, again, slightly worse outcome, but there had been some other co-founding, and I know there was a follow-up Italian paper that just came and said the question, the significant or the role of the blood group if used as a biomarker, and it's currently has not been recommended to use it as a biomarker in predicting the severity. The next paper that we worked with the RCSI, again, this is a fantastic paper. You're all aware of what happened here in Bahrain in September. You remember the big wave we had. Just remind you that was during the Eid holidays, and that was during the Ashura period. Again, the question was simply, do we think it's actually the spread of the virus is related to the behavior of the individuals or not? And you all know during the religious holidays, especially here in Bahrain, it is a group of a big family that they tend to gather, and we had massive number of cases. And again, there are other supportive evidence that it's the behavior of individual that linked to the spread of the virus rather than other factors. And that has been submitted to the BMJ for publication. I'm going to spend time talking about the randomization of convalescent plasma in the next slide. But we have done also the antigen rapid test. We have over 9,800 data. This particular paper, we talked about those who have been visited BDF with symptoms, and we just correlate first of all the utility of antigen with the PCR, and then we moved on and we looked at the nasopharyngeal and the nasal. Again, the rationale we have done this, and again, you all know now nasal swab has been accepted, but back then nasal swab was not being accepted as a specimen to be used. But we have been shown with good amount of data that if you use a proper nasal swab, it's as good as nasopharyngeal in screening for COVID-19. And now most of the rapid antigen test company, they have granted the FDA approval in using the nasal swab. Remind you, nasal swab is less invasive. You can use it in kids, and it will be even self-collected much easier than the nasopharyngeal. Next is the diagnostic and monitoring utility of saliva. Again, this was in collaboration with the RCSI students. And another amazing paper that's been submitted to the Journal of Clinical Virology. I think Saad was with us in this working group. We actually looked at the utility of the saliva. If nasal was good, the question was, can you detect some nucleic acid on the saliva, which will be much more convenient for those who are unable to use the nasopharyngeal due to a surgical, for example, procedure, or for children. And again, we have shown the diagnostic accuracy if you use the saliva. However, you have to use it in a symptomatic individual, and their symptoms has to be within 7 days and not longer than that. And again, there was another publication supported this from Kuwait that showed the utility of saliva. And again, now saliva has been used as an alternative method in collecting the specimen. We have also looked at the association between CT values and predicting. This is one of the exciting modeling paper that was in collaboration with RCSI and the AGU. Again, I really encourage you to see it at the preprint. It's at the Medical Archive. Beautiful paper submitted to the epidemic. The question was, can viral load predict as a model in a cluster of cases? Again, we have seen a lot of cases when you had high clusters outbreak, it tend to have a lower CT value. Lower CT value indicating a higher viral load. It indicates the cycle threshold. Again, it's a very lengthy paper, a bit complex. I would encourage you, if you're really interested in modeling, this is an excellent paper to go. And this was actually also, we worked with Dr. Michael Mina. Probably a few of you know him. He's one of the pioneer in epidemiology from Harvard University. And then the last paper that has been submitted to the British Journal, again, this is a fantastic paper, great collaboration from RCSI and from AGU students and from staff from RCSI. When we ask the question, is sickle cell disease is a risk factor for COVID-19? This is the largest multi-center national respective cohort trial. I think that was at 2000, where we have looked at 380 something individual that they had sickle cell disease. The bottom line, again, proven to the first paper, that was not a risk factor. Sickle cell disease was not a risk factor to have a complicated sickle cell disease or a direct causation. Next slide. Those are the 13 ongoing research that purely we are working with the RCSI. Very proud and very happy to see this huge interest from the RCSI in supporting all of this. As I said, I can think of a country that we have worked in a very busy day with the COVID-19, and they have the interest in research. But those are very interesting research topics. Again, if some of you still has the interest, we have actually some group has been joined or lined up for this. If you are one of those who wish to be a member, we'll be delighted to have you on board. But again, lots of emphasis about the use of antigen tests. Bahrain has a lot of data about the clinical use of antigen. We have shown this in several locations like schools. We have been using at the hospital now. In the community, we have huge investment in the community. The whole idea, we wanted the community now to engage with us and to reduce the burden of use antigen. I can spend the whole day talking about the antigen. There are so many things to say about it, but just for the sake of the time, I think I have to end with this, Khawla.
Thank you very much, Professor Qahtani, for this very informative talk. And just a reminder for all our audience to submit your questions in the Q&A or the chat box at the right corner of your screen. Next slide, please. Our next guest speaker is Dr. Abdul Karim Abdul Rahman, a resident at Mohammed bin Khalifa Cardiac Center and a member of the National Coronavirus COVID-19 Research Group, who is presenting his research on the efficacy and safety of hydroxychloroquine in COVID-19 patients.
Hello, good morning. Thank you, Khawla. It's a pleasure to be here, and I would like to take this opportunity to thank RCSI Group, namely Professor Stephen, Celine, Fawzia, Nevin, and Hassan, who have been working backstage to organize and host this symposium, which highlights the important research that was done in Kingdom of Bahrain to fight this horrible virus. And moreover, I would also like, it gives me such a great privilege to be speaking directly after Professor Sheikh Khalid and my mentor, Professor Dr. Untitled Qahtani, who I personally have learned a lot from over the past few years, and especially during this pandemic. And I was so lucky to be working alongside him for the past 10 months. I'll be presenting a manuscript that we recently published. It's entitled The Efficacy and Safety of Hydroxychloroquine in COVID-19 Patients. Next slide, please. So this research was actually a collaborative teamwork from different people and different residents and RCSI research students and RCSI faculty. And would like to take this opportunity also to thank each and one of them, as they have contributed greatly to the success of this research. Next slide, please. We have published the preprint of this research back in November in the Medical Archives, and we recently, next slide please. We recently received this email that I'm happy to share with you from the Infectious Disease and Therapy Journal when they accepted our manuscript recently earlier this week. It's always good to have this email, so I wanted to share it with you. Next slide please. So why did we do this research? We wanted to answer the important clinical question: does hydroxychloroquine treatment have an effect on COVID-19 outcomes in COVID-19 patients in Bahrain? And why we asked this question, it's related to a series of timely events that I'm going to walk you through. Next slide, please. So back in March 2020, and this is a screenshot from the treatment protocol of the national protocol, the first edition in March. And we can see that hydroxychloroquine have been used frequently to treat pneumonias and also uncomplicated infections for people with risk factors. Next slide, please. And that time it was based on 3 main published researches at that time. The first one was published in February in a journal of cell research, and it showed that it was an in vitro study which showed that chloroquine effectively inhibited SARS-CoV-2. Next slide, please. Then another research, which was also an in vitro research, which was published in the Cell Infectious Diseases journal, and it showed that hydroxychloroquine was even a more potent inhibitor of SARS-CoV-2. And this was actually very reassuring to people and physicians, because physicians tend to have a good experience with this drug, as it has been used frequently in rheumatology clinics. Next slide, please. And then another paper, and it was published at that time as a preprint. It was a trial that was done in France on a small sample of patients and published in the International Journal of Antimicrobial Agents, which showed that hydroxychloroquine treatment was significantly associated with viral load reduction and disappearance. And this also enforced and increased the evidence related to using hydroxychloroquine in COVID-19 patients. Next slide, please. So in Bahrain, they started using it back in March. We started using it back in March, and not just Bahrain. Moreover, other countries like India, Brazil, UAE, KSA, and even the USA started using it. Moreover, next slide, please. The FDA have issued an emergency use authorization to use the same drug in COVID-19 patients and on the 27th of April. Next slide, please. However, soon in May, the first paper was published in the New England Journal of Medicine regarding hydroxychloroquine in hospitalized COVID-19 patients, and it showed, on the contrary, that hydroxychloroquine was not associated with a greatly lowered or increased risk of intubation or death. Next slide, please. Another paper which was published in the JAMA based on an observational study in New York, it showed that hydroxychloroquine was not beneficial in COVID-19 patients, and moreover, it showed that hydroxychloroquine increased the risk of cardiac arrest and was harmful. And this captured the attention of many people worldwide. And soon the WHO discontinued the use of hydroxychloroquine in its Solidarity clinical trials. Next slide, please. The FDA then also stopped and revoked the EUA authorization for hydroxychloroquine in COVID-19. Next slide, please. And based on those scientific evidence, we also removed it from the national protocol. And it coincided at the same time, if you can see from these graphs, with an increase in active cases and an increase in deaths. Next slide, please. Hence, many people and healthcare workers started associating these two events together about withholding hydroxychloroquine and the increase in cases and deaths. And this caused the confusion within the medical community, especially in Bahrain. And moreover, also some patients started asking for the medication when they were admitted, and a proportion of physicians continued to prescribe it. Next slide, please. So this is what we did. So we decided to do a retrospective cohort to study the exact effect of hydroxychloroquine in the admitted COVID-19 patients. Next slide, please. And this is how we did it. So we took 4 of the main Ministry of Health centers, and we took all the patients with COVID-19 who've been admitted there. And we randomly selected patients and stratified them based on the prescription and receiving hydroxychloroquine. We then selected a random sample from these 2 groups, and we extracted their records, medical records, and went through them and gathered the relevant information. And we studied 3 main outcomes, with a primary outcome, which was the requirement of ventilation, whether non-invasive or invasive ventilation, or death as a primary outcome. We also studied safety outcomes related to cardiac arrhythmias, QT prolongation, diarrhea, and hypoglycemia. We also studied a secondary outcome based on the length of stay in days for survivors. Next slide, please. Based on the research methodology and the research design, which was very amenable to confounding factors, so we decided to go with 3 different methods to control for confounding in this paper. And based on our belief that using 3 different methods, and if they coincide and give us similar results, we'd have more confidence in our results and in our interpretation and inferences. So we use a multivariable logistic regression model as our primary analysis. We also use propensity score based analysis as a matched analysis, and also as a covariate in the regression model. Moreover, in the same study, we also tested for effect modification for the course prescription of azithromycin, which also gained attention of many physicians. So we studied that as an effect modifier. Moreover, also we studied the baseline severity of disease, whether the patient were hypoxic or not. Next slide, please. So we reviewed a total of 1,849 cases, and we had a few of them excluded, and we had a total of 1,571 cases included in our study. Next slide, please. We had 440 patients on hydroxychloroquine, and the rest were in the control group. Next slide, please. Let's just see the table. So we can see that we had 440 patients in the hydroxychloroquine group, and these patients had older age, 53.4 compared to 44.6. There were more Bahrainis in the hydroxychloroquine group, and comorbidities were also even more, namely diabetes and hypertension. Moreover, as this is only part of table 1, the hydroxychloroquine group also required more supplemental oxygen on admission, and they had more diagnoses of pneumonia on admission. Next slide, please. So this kept the hydroxychloroquine group at a higher baseline risk throughout their admission. And as so, we expected that hydroxychloroquine patients had a higher outcome of the composite endpoint. So 5.5% of the hydroxychloroquine patients developed the outcome, and only 3.9 patients who were not on hydroxychloroquine developed the outcome. And this was basically based upon the baseline risk of the patients. So when we adjusted for confounders, we found that there was no effect of hydroxychloroquine on the primary outcome studied through the use of 3 different methods, as you can see that all the confidence intervals crosses 1. Moreover, when we tested for effect modification and stratified patients based on co-prescription of azithromycin or if they were hypoxic or not on admission, again, hydroxychloroquine did not affect the outcome. Next slide, please. So despite the significant differences between the 2 groups, the older age, the more comorbidities, the higher baseline risk, we still had no clinical benefit from using hydroxychloroquine in COVID-19 patients. Next slide, please. The secondary outcome, which was the length of stay, we found differences based on a crude analysis using t-tests that hydroxychloroquine had a higher length of stay because of their higher baseline risk when compared to the control group. However, after adjusting for confounding factors, we saw that hydroxychloroquine did not affect the length of hospitalization in survivor patients. Next slide, please. With regard to safety outcomes and adverse events, we found very interesting results. Namely, hypoglycemia and diarrhea were significantly associated with the intake of hydroxychloroquine. And this was very interesting because hypoglycemia could actually be a very important risk, and it could be an adverse event, especially when prescribed out of hospital settings. Moreover, a study was also published previously that patients who are diabetic and who receive hydroxychloroquine could have a decreased insulin units required, as hydroxychloroquine can induce hypoglycemia. Diarrhea is also a known adverse event of hydroxychloroquine and have been reported in other studies. It was very interesting to find that QT prolongation was not significant with the prescription of hydroxychloroquine. And this is on the contrary to many papers, because hydroxychloroquine was usually associated with QT prolongation, which led to cardiac arrhythmias in other reported research. However, we believe that this insignificant finding was due to the local protocol used in our hospitals. So basically, patients who were on hydroxychloroquine were observed more for QT prolongation. All of them had daily ECGs done. And whenever a patient exceeded a QT of 470, they were usually withheld from the medication. And moreover, patients with cardiac events and with a history of cardiac events or increased risk of cardiac arrhythmias were usually not prescribed hydroxychloroquine. Anyways, both QT prolongation and cardiac arrhythmias, after controlling for confounding, they were not significant. Next slide, please. Our research came into agreement with many of the big studies that were published regarding hydroxychloroquine. Mainly, and the most important one was the recovery trial, which was done in the UK, and which were the results published in the New England Journal of Medicine, which showed no benefit in decreasing mortality or invasive nor ventilation in patients taking hydroxychloroquine. And this was done on more than 4,716 patients across 176 hospitals in a double-blinded randomized controlled trial. Next slide, please. And another trial which was conducted in the US on 479 patients, and the results was published in JAMA, also showed similar results, and hydroxychloroquine did not improve clinical outcomes in patients with COVID-19 respiratory illness. Next slide, please. And another study in Brazil, which was another randomized controlled clinical trial published in the New England Journal of Medicine, also agreed with our findings. Next slide, please. And lastly, another study based on New York and published in JAMA also showed very, very similar findings. So this all gave us much more confidence in our results and our interpretation that our study was a high quality observational study which matched the results and inferences done in other larger observational studies and randomized controlled trials. So next slide, please. So in conclusion, our results showed no significant beneficial effect from using hydroxychloroquine on the outcome of COVID-19 patients. And we also highlighted that the risk of hypoglycemia could be serious, especially when used as an outpatient. And thank you.
Thank you, Dr. Abdul Karim, for your presentation. Next slide, please. Our final guest speaker for today is Mr. Saad Mallah, a third-year medical student and student researcher at RCSI Bahrain, who has been supporting the National Coronavirus COVID-19 Research Group. He's presenting COVID-19 Year 1, Breaking Down a Global Health Crisis.
Thank you, Dr. Khalid, for the introduction. I'm very honored to be presenting here today in front of many of the esteemed guests joining us. So I will be presenting what, as the title suggests, a breakdown of the global health crisis. So I was asked to present this project specifically, I believe, because it had a lot of students involved in it, and a lot of RCSI, very multinational and multidisciplinary collaborations. So this was a review paper, more appropriately maybe a review booklet, which I'll get into now. So next slide, please. So our task was very straightforward. We started out around March, which was the beginning of the COVID-19 pandemic, or at least the beginning of all the global changes that were taking place. And the project took around a duration of around 6 months to complete. So our main aim was to synthesize the global impact of the virus from a micro to a macro scale. And that was something that we believe made this project unique, in the sense that we didn't just focus on, as most papers do, on just, for example, the microbiology of the virus or just the global health, but we wanted this to really be a very comprehensive project. So we wanted to disseminate very evidence-based and up-to-date information about COVID-19, and not just for medical communities, but also for members of the public. And I think COVID-19 has shown us that we can no longer discuss health and medicine simply within us as healthcare professionals, but it has shown us the really the large involvement of the patients and the public in these diseases. And so we also wanted this to be something for current and future reference. So as medical students, this is not, we don't always get the opportunity to be involved to witness the making of a new disease. So the idea that hopefully in the coming years, people would be going back to these papers that we're publishing and this data, and be able to reference a disease that we were able to contribute to the knowledge of. Next slide, please. So as I was saying, this was a truly multinational and multidisciplinary team. So we had around 6 or 7 medical students working on this project. We also had a political science, public policy, and international relations student from the University of Toronto to help offer more of the economic burdens and the global public health, global affairs perspective. We also had pharmacotherapy specialists, both in cardiology and infectious diseases, from Cleveland Clinic in Abu Dhabi. And their role obviously was very essential because, well, first of all, because of a lot of the discussions about the novel potential therapies, and also cardiology because of it was a center of discussion and remains to be a center of discussion. And of course, we had an infectious disease and infection control and really COVID-19 expert, Dr. Manaf, with us. Next slide, please. So the content, as you can see from here, was to cover everything we can about the virus. So we started out giving an introduction about the epidemiology of the disease, and then moving on to a micro scale, discussing the virulence, some of the things that Dr. Stephen presented in the beginning of his presentation about the cell entry, cellular replication. We discussed the pathogenesis and the risk factors that may have increased the chance of severe disease, the transmissions and the precautions and the screening and diagnosis of COVID-19. We then moved into more of a clinical aspect of the disease, discussing patient management, specifically in children and pregnant females, and of course, cardiovascular controversies. We moved on to vaccination efforts and novel potential therapies. And as Dr. Abdel Karim mentioned in his presentation, some of these medications, like hydroxychloroquine, were the forefront in treating COVID-19, and then before we know it, they were recommended against. And so that was a challenge as well in coming up and writing this document because of the ever-changing information. Every day there was a new paper being published that suggested the exact opposite of the current understanding. And finally, we moved on to more of a macro. So as you can see, we're getting more of the macro approach now with discussions on the global health responses, and we divided that by region. So all the different regions and all the different responses. And finally, a global economic burden discussion, which was something that we believe made our paper unique because as public health professionals, as medical professionals, and even as members of the public, COVID-19 has also shown us the idea of One Health, the idea that everything is interconnected, whether as patients or and public health and disease. Next slide, please. So the end product was a 60-page all-encompassing booklet. It was around 15,000 words and around 300 references, which we systematically have gone through. The paper is currently under review by the Annals of Clinical Microbiology and Antimicrobials, which is published by BMC Springer Nature, and thankfully has passed the editor stage and is currently under review. So next slide, please. So I just wanted to share some of the figures that we used. So in this paper, we focused a lot on illustrations. We designed our own illustrations. Every figure involved or used in our paper was something we've designed. And the reasons for that is because obviously illustrations were always the best way to disseminate information, and also for members of the public, because for them, simplifying the disease in these illustrations really helped. So this one was about epidemiology, and as you can see, now this is continuously updated. So this is the older one that ended in September. So as you can see, there was an exponential rise in the number of COVID-19 cases. When we started around in February with 85,000 cases, it took around 3 months to get to 3 million, but then only 1 month to get to 6 million, and only another month to double that, and so on. So it was truly an exponential rise in terms of cases. Next slide, please. So this figure here, we used, what we did here is that we used data from multiple sources to create some of these figures. So in an ideal controlled environment, what you would see here is the percentage of the world population that a certain continent contributed to would also be equal to the percentage of COVID-19 cases it would contribute to. So the blue and orange lines would both be equal in a controlled environment. Now, obviously, that was not the case. So as you can see, South America and North America had a disproportionate number of COVID-19 cases when compared to, for example, Asia and Africa, who did quite well. And specifically Africa, and I will get back to Africa further on. Europe didn't do as well. And obviously Asia included the Middle East. So that was nice to see. Next slide, please. So this was one of the illustrations where we showed the overall complications of COVID-19. So we used tens of case reports, cohort studies, anything we can get our hands on to see the true impact of pathogenesis and complications of COVID-19. So as you can see, this is, from a medical perspective, a fascinating disease, and obviously from a patient perspective, a very scary one, because in the beginning we understood COVID-19 to be a respiratory disease. Then soon after, we moved on to a lot of the cardiac and hematologic as well as vascular pathologies that were a hot topic. And then later on, throughout, there was a lot of systemic discussions about the impact of the disease. And then some cutaneous findings. Some of them were called, for example, COVID-19 toes, which were some findings that some researchers were identifying and physicians. And then finally, more recently, a lot of neurologic complications in COVID-19. And we actually have, we're working on a number of new reviews now discussing some of the neurological aspects and breaking them down in COVID-19. So next slide, please. So in this slide here, we discussed 5 countries that had the highest number of cases per million in the first 30 days since their first confirmed case. So we used that 1 month period because that 1 month period is a critical time period. When you have as a country your first infectious case, the precautions that the country takes in that 1 month period is critical to the further development of the disease. So you can see here that the countries in red had the worst cases per million, the highest cases per million, and the countries in green had the lowest cases per million. And you can see how the countries in red also had the highest case fatality rates. So there was a correlation, it seems, between the case fatality rate and the number of cases, probably because of the overwhelming of the healthcare system and the infrastructure because of the increased number of cases. And we saw that specifically in Italy, which had its infrastructure and healthcare system overwhelmed. Next slide, please. So this was probably remains one of my favorite figures. And what we did with this one here is that we used data from the Oxford Government Response Tracker to find out to choose 5 specific measures. So cancellation of public events, school closure, work closure, international travel restrictions, and domestic travel restrictions. And what we did is that we wanted to see at what day did these countries take the initiatives mentioned. And then we took that date and we found out how many cases per million the countries had on that day. So that way we standardized the playing field, because you can't compare one country to another in terms of their public health measures, because each country had their first infectious case at a different time. So here, when we standardized that, you can see beautifully that these green countries here, had taken the actions, the public health measures, around 0 cases per million. In fact, some of these countries closed down even before they had their first infectious case. And a result of that, clearly, was that they had the lowest number of cases per million in the first 1 month since their first case. Compare that to, for example, Portugal, who only closed work when they had 1,600 cases per million, and only restricted domestic travel in around 1,300 cases per million. And they had some of the worst and largest outbreaks in their countries. Next slide. Oh, one more thing, actually. So here, we excluded countries that had a population of less than 1 million, and we also excluded countries who had a civil war, specifically Syria, because we believe there was a lot of confoundings and extrapolation involved. Next slide, please. So this is exactly the previous figure. This was not part of our paper, but this was a previous draft that we removed. And the reason I'm including this is because we actually had a third exclusion criteria. We excluded the continent of Africa at the beginning because we were under the impression, as well as many other researchers and the public, we were under the impression that Africa did not have a large number of cases simply because they did not have a lot of travel. But a lot of social media posts and a lot of news articles started showing that that was not actually the case, and that there was a lot of bias involved. The African CDC, at the end of the day, is one of maybe the most equipped in dealing with and the most experienced in dealing with infectious diseases. And so we decided to include Africa and see if the data changed. And as you can see, and as you saw from the previous slide, these countries took, the African countries took very serious and very successful public health measures very early on in the pandemic. So I just wanted to note this out because bias in research really sometimes can be involved in ways that we may not identify originally. And I think it's very important that we always try to educate ourselves as much as possible so that we don't victimize others of that bias, because specifically underestimating of, for example, the continent of Africa is something that we suffer from here as well, a lot from the Middle East. We a lot of the times we are victims of that underestimation type of perception, when in reality, as this talk and this symposium shows, Bahrain, for example, has been a world leader in their response against COVID-19. So I think it's very important that we always keep that in mind. Next slide, please. So yeah, thank you a lot, and I also wanted to just end with a special thank you for Dr. Manaf. He has truly been empowering and enabling the students, and what has shown us that he has been a great visionary as well for us. And Dr. Abdul Karim as well, for being a great mentor and a very patient mentor. I've learned a lot during these past 6 months working with the task force, and I look forward to everything that we'll be achieving together. Thank you.
Thank you, Mr. Mallah, for the presentation. And this really concludes our presentations on the collaborative research on COVID-19 held in the Kingdom of Bahrain. Next slide, please. Now I would like to proceed with the Q&A session, and I do see that there are a couple of questions in the chat box. So the first question is probably directed to Professor Manaf, and the question is, do we have any data for G6PD and COVID-19?
No, in fact, we don't have any collected data in Bahrain about G6PD.
Okay. Thank you, Prof. The next question is from Prof. Nuha, and I believe it's also directed to Professor Manaf. And the question is, having implemented the brilliant Be Aware application, do we have any studies on how this facilitated control of the epidemic, the pandemic, and what impact it had?
So that's an excellent question. Actually never came to our attention about using the utility of the Be Aware application and the benefit. Something we're going to keep it in mind. Thank you.
Thank you, Professor Manaf. And I do invite all the attendees to submit their questions again at the chat box on the right corner. I do have personally a question also for Professor Manaf, and then a question for Abdul Karim. You know, the volume of research we saw Professor Manaf done in such a short period of time is pretty remarkable. What is our plan? And I think a lot of it stems from the fact that there's this centralized research group working in collaboration and sort of easing the process of research rather than working independently as previously pre-COVID-19. So what I'm interested in is, what are our plans going forward post the COVID-19, hopefully pandemic, and hopefully soon, in terms of also, you know, plans to centralize research in Bahrain to make it easier to collect data from multi-centers and continue this progress that we've witnessed?
So I think the first of all, we would like to see most of our policies here in Bahrain that is related to our healthcare. It's actually been related to our own data, and I think that was the main intention from this research group. Every single policy that's related to COVID-19, you have a reference from your own data, and I think sharing those data earlier on, even with the international community, has put Bahrain into a position where we have been granted to have the vaccination trial. We have done three biggest randomized controlled trial, the plasma and the favipiravir trial, and we have also done the vaccination, which is the Sinopharm double-blinded control trial. I think it's an opportunity where we have a young researcher, like for example Saad and other colleagues. I think we have tremendous efforts from our students. They just need some guidance. They need maybe support from the college as well. There are some brilliant ideas. I think some of the RCs student has them, creating a networking research group, not necessarily just for RCSI, but maybe a joint effort with other colleges. This COVID-19 brought up so many ideas. We just need the time to sit and put them all together, and I'm really hopeful that in the very near future, Bahrain gonna be will be a leading in terms of the research, if not regionally, hopefully will be internationally.
Brilliant, thank you, Professor. The other question I have is or actually let me move on to the audience's questions. To give people the chance. So I have a question here from Dr. Nigel Stevenson. And the question is, thanks for the excellent presentations. Bahrain has really led the way internationally in combating this virus. The vaccine rollout is particularly impressive. Will there be research on, one, the protective effectiveness i.e. antibody levels of the vaccines in Bahrain. That's the first part. And the second part, the effect of long COVID in Bahrain.
Yeah, I can take that. Thank you, Nigel, for your questions. Yes, I do remember from the vaccine trial that we have done with the Sinopharm. We did a parallel study. We do have the antibody results of those trials. During the emergency authorization as well, we have done another trial where we have collected all the serology blood sample. The interesting part, Nigel, is that in Bahrain, we do have now the neutralizing antibodies. This is something that we introduced back in September. That's why the plasma paper, the one I did not present, we had some technicality in terms of having the result of the neutralizing antibody. But now with this technology being available here in Bahrain, this is non-culture based neutralizing antibodies. I do understand if we have the culture-based one, that would be the more reliable one. But at this stage, we do have a good amount of blood sample from our volunteers that really showed an excellent efficacy for the inactivated vaccine. We have not implemented this for the messenger mRNA yet, but there are so many things, as you know, that you can do with the COVID vaccination and the antibody level as well.
Thank you, Professor Manaf. I'll direct a question now to Professor Stephen Atkins. And the question is, Prof, if we do have a vaccine available or vaccines available right now for COVID-19, what is really the point now of the favipiravir trial?
Thanks, Carla. That's a very good question indeed. And of course, what we've actually seen is that we don't know yet whether or not those people who are vaccinated or will be fully protected. You've probably seen in the news, quite a few people have complained of catching COVID-19 disease when in fact they have actually had the vaccine. So there's always going to be a requirement, I suspect, for having efficacious medication to actually treat those people, even if they're vaccinated, but may have a kind of a milder disease profile. So there's always going to be the need for, and also for those, of course, who don't get vaccinated. Thank you.
Brilliant, thank you, Prof. Another question submitted for Professor Manaf, and the question is about public behavior and willingness to vaccinate. The question is it possible that the belief of getting vaccinated just with the first dose, rather than the 2 dosages, is there any data to suggest that there are false beliefs about getting complete immunity with just 1 dose? And is that evident, even though it's probably early to say, of low compliance to a second dose vaccination or 2-dose vaccination rather than 1?
So from the Bahrain data, so if I go to the volunteers and maybe going to the real life vaccination program, so it's definitely with the volunteers, maybe because it was a trial where there's a strict follow-up, we did not have that behavior. With the real vaccination candidates, we did face that, not with the first dose, but with having the 2 doses, having the impression this would be effective and they should not follow the precautionary measures. And we have several times have said that in our conferences that we really need to reach up to 70% of the population to be vaccinated. And that's what we call the herd immunity before we lift up any precautionary measures. Having said that, we have been looking at the behavior of a healthcare worker. We are working with a group from the United States, Abdul Karim also with me on that, where we have sent a questionnaire. We reached, I think, above 1,000 healthcare workers in Bahrain, set of questionnaires about their belief and their disbelief about COVID-19 vaccines. I don't have the result yet, but I'm happy to say that another student from RCSI are helping us on this research. This is an international research collaborative where we're going to have the result very soon.
Very good. Thank you, Professor Manaf. While you're on the line as well, another question. Do we have any insights into the presence or absence of the new variants of COVID-19, such as the UK B117 or South African B1351 in Bahrain? So really rather a general COVID-19 public question.
Yes. So just to remind the audience, this the new variant strain has been circulating or have been known since September. So this is not really a new strain. Nothing to be worried about it except from the transmission. Luckily enough, all the clinical scenarios that have been affected in UK or South Africa showed that did not change the severity of the disease, and I think that's very important. Does it have really link to the virus itself being transmissible, or really the behavior again of the individuals? We all know during this time of the season, it was the family gathering during the holiday season, and I do believe that has a huge impact on it. We are looking for that. We're screening those who are coming from those countries for the new variant. We have an excellent collaboration even with the UAE as well between us. So until that time, we don't have any such a case, and I think if we do have, we're going to report that immediately.
Very good, thank you, Prof. And again, inviting the attendees to submit their questions in the Q&A box. I have a question now for Dr. Abdul Karim. We've done recently a clinical trial on hydroxychloroquine and autologous islet transplantation, so outside of COVID-19. And interestingly, we saw an increased rate of Clostridium difficile, C. diff, as a cause of diarrhea in patients receiving hydroxychloroquine. My question is there any evidence of increased C. diff in patients receiving HCQ or hydroxychloroquine? Or is this a non-infectious diarrhea related to the pharmacological properties of the drug?
Well, that's actually very interesting because the diarrhea that we had could be infectious or non-infectious. However, one of the limitations is that we didn't have access to laboratory and or microbiological data at that time. So we don't have enough data to say if what was infectious or non-infectious. However, it's something that we can retrospectively look again, and I think it would be an interesting finding to see so.
Thank you very much. I'm going to move on now to Mr. Saad. And my question to you, and probably there are a few students also in the audience attending, is I want you to talk to us about the biggest challenges that you faced in such a research project, and what advice would you give any medical student attendees for furthering their research efforts in the future?
Thank you, Dr. Khadas. So specifically with the review paper that I mentioned, I think our biggest challenge was the fact that we were doing an interim review. Most review papers that are usually written retrospectively completely once a pandemic is over. But this interim review, one of the biggest challenges was the fact that every single day there were thousands of new papers that we had to review, and sometimes completely changing the whole discussion several times. And obviously, always working with large groups of people, there's always difficulties in trying to put it all together. So I think that was one of the biggest challenges with that specific project. Generally, for research and the other projects that I was involved with the task force with, I think I've seen a lot of students always approach and ask about research and try to get these opportunities, and I think that's always the first step. There's sometimes this preconception that research is something that is far off and far away from students and something that they're not capable of initiating. But that's actually not true. A lot of the times, a lot of these RCSI staff members and other professors are very much willing to have students involved with them and mentor them and guide them. And just generally, even a lot of projects students can initiate, and I've seen that students have initiated projects themselves. They've written papers or perspectives or or even review papers, and then approached professors with them. And I think that's always the start. And that's how it started for me as well. I started out with the review, approached Dr. Manaf, and then eventually started working, continued working with the team on other projects. So I think it's always that, just always pursuing the opportunities, and just doors continue to open.
Thank you. Thank you, Saad, for the insight, and hopefully that will motivate our students to pursue more projects and to join really the other COVID-19 projects for the experience, for the publication. So I don't see any more questions in the Q&A box. If anyone has any questions in the final few minutes. I think otherwise we will conclude. So thank you everyone for joining. This concludes our online symposium on the collaborative research on COVID-19 in the Kingdom of Bahrain, held under the patronage of His Excellency Lieutenant General Dr. Sheikh Mohammed bin Abdullah Al Khalifa, Chairman of the Supreme Council of Health. This symposium was held in collaboration with the National Task Force for Combating the Coronavirus COVID-19 and the National Health Regulatory Authority and RCSI Bahrain. Next slide, please. Okay, I'd like to thank our guest of honor, His Excellency Major General Professor Sheikh Khalid bin Ali Al Khalifa, Director of the Royal Medical Services and Chair of the Research Committee of the National Task Force for Combating the Coronavirus COVID-19. I'd like to also thank our guest speakers for their valuable contributions. Professor Stephen Atkin, Head of Postgraduate Studies and Research at RCSI Bahrain, Prof. Manaf Al-Qahtani, Infectious Disease Consultant and Microbiologist at the Bahrain Defense Force Hospital and member of the National Task Force for Combating the Coronavirus COVID-19, Dr. Abdul Karim Abdul Rahman, resident at Mohammed bin Khalifa Cardiac Center and a member of the National Coronavirus COVID-19 Research Group. And finally, Mr. Saad Mallah, RCSI third-year medical student. I'd like to thank you, our attendees, for your participation. Please stay safe and follow the COVID-19 safety measures. I wish you a good day.